The cell cycle includes interphase, mitosis, and cytokinesis. Interphase, which takes up most of the cell cycle, is broken into three phases: G1, a growth period for the cell, S, during which the DNA of the cell is duplicated, and G2, during which the cell grows, differentiates, and prepares for mitosis. By the beginning of mitosis, the cell's DNA has been duplicated, but is not condensed from chromatin into chromosomes. The nuclear envelope surrounds the nucleus, and the nucleus contains one or more nuclei, and the centrosome has duplicated. During prophase, the first phase of mitosis, chromatin condenses into chromosomes. The nucleoli disappear. Each duplicated chromosome appears as two identical sister chromatids joined at their centromeres. The mitotic spindle, composed of the centrosomes and microtubules that extend from them, begins to form. Radial arrays of shorter microtubules that extend from the centrosomes are called asters. Centrosomes move away from each other, partially pushed by the lengthening microtubules between them. During prometaphase, the nuclear envelope begins to break apart. Microtubules extending from each centrosome can now enter the nuclear area. The chromosomes condense even further. Each of the two chromatids, every chromosome now has a specialized protein structure at the centromere. Some of the microtubules attach to the kinetochores, becoming kinetochore microtubules. Non-kinetochore microtubules interact with those from the opposite pole of the spindle. During metaphase, the centrosomes now move to the opposite poles of the cell. The kinetochores of the sister chromatids are attached to kinetochore microtubules coming from opposite poles. The centrosomes line up along the metaphase plate, a plane that is equidistant from the spindle's two poles. During anaphase, the shortest phase of mitosis, cohesins are cleaved, allowing all sister chromatids to separate, each becoming a chromosome. The daughter chromosomes are pulled to opposite poles as the kinetochore microtubules shorten. The cell elongates as non-kinetochore microtubules lengthen. Both ends of the cell now have a full set of chromosomes. During telophase, the nucleoli reappear. The nuclear envelopes reform from the fragments of the parent cell's nuclear envelope and parts of the endomembrane system. Chromosomes have become less condensed. Any remaining spindle microtubules depolymerize, and mitosis is complete. After mitosis, cytokinesis splits the cell into two daughter cells. In animal cells, the cleavage furrow forms, pinching the cell into two daughter cells. At the end of mitosis, two daughter cells have formed, which are genetically identical to their parent, with an equal number of chromosomes with the parent and each other. Mitosis is important because it allows some organisms to reproduce asexually and allows multicellular organisms to grow and to replace lost or damaged cells. Meiosis is another type of cell division that is important because it allows organisms to reproduce sexually and ensures genetically diverse offspring. Meiosis 1 begins with prophase 1, during which chromosomes begin to condense and homologous chromosomes pair up by length and become physically connected along their lengths, entering a state called synapsis. Crossing over occurs while chromosomes are in synapsis. Corresponding segments of DNA switch places on homologous chromosomes. After synapsis, chromosomes in each pair have separated slightly. Each homologous pair has one or more chiasmata, an X-shaped region where crossing over has occurred. As in mitosis, centrosome movement, spindle formation, and nuclear envelope breakdown occur. In metaphase 2, pairs of homologous chromosomes are now arranged at the metaphase plate, with one chromosome in each pair facing each pole. Homologs are attached to microtubules from opposite poles. Phase 1 begins with the breakdown of proteins responsible for sister chromatid cohesion along chromatid arms, allowing homologs to separate. Homologs are guided to opposite poles by the spindle apparatus. Sister chromatids move together to the same pole. During telophase 1 and cytokinesis, each half of the cell has a full haploid set of duplicated chromosomes. Cytokinesis usually occurs at the same time as telophase 1, forming two haploid daughter cells. No chromosome duplication occurs between meiosis 1 and 2. Meiosis 2 begins with prophase 2. A spindle apparatus forms. Chromosomes move towards the metaphase 2 plate. In metaphase 2, the chromosomes align along the metaphase plate as in mitosis. Because of crossing over in prophase 1, the chromatids of each chromosome are not genetically identical. Kinetochores of sister chromatids attach to microtubules from opposite poles. During anaphase 2, sister chromatids separate, becoming individual chromosomes, and move to opposite poles. During telophase 2 and cytokinesis, nuclei form. Chromosomes begin to decondense and cytokinesis occurs. Meiosis results in 4 haploid daughter cells. DNA replication for both mitosis and meiosis occurs during interphase before cell division. Mitosis has only one round of cell divisions, while meiosis has two. Synapsis of homologous chromosomes only occurs during meiosis. The result of mitosis is two diploid daughter cells that are genetically identical to the parent cell, and the result of meiosis is four haploid daughter cells that are genetically diverse from each other and the parent.